everyone. Welcome to episode 123 of Cell and Gene, the podcast. I'm Erin Harris, your host, and I think you'll find my conversation with Dr. Panteli Theocharous important and timely. He and I dive into exactly where cell therapy trials create the most friction for patients today, and the practical fixes that can make them faster, clearer, and more humane. Panteli is a true heavyweight in cell and gene therapy with 35 years bridging clinical hematology at the Royal Free Hospital and UCL, executive leadership at Thermo Fisher and PPD, where he chaired their CGT Institute. He held CMO roles at Lift Biosciences and CTI, and most recently, Chief Therapeutics Officer at Stratus Therapeutics, helping secure a $55 million Series A1 raise. Panteli's the rare expert who's not only shaped CAR-T, AAV, and stem cell strategies at scale, but also champions the patient journey from trial design through long-term follow-up. He's endlessly knowledgeable, and you'll hear in his voice just how much he cares about the patient journey. And so without further ado, here is my conversation with Dr. Panteli Theocharous. Enjoy. Panteli, hi, welcome to Cell and Gene, the podcast. I am so happy you're here. You and I met back in November of 2025 at Cell 2025 back in London. And we talked about perhaps a way we can work together and what would make sense for Cell and Gene's audience and Cell and Gene, the podcast's listeners. And I'm just so thrilled we were able to work a way out to bring you onto the podcast and put your tremendous insight in front of our listeners. So I can't thank you enough for your time. And I'm so glad you're here.
Erin, look, it's a real pleasure to be here. Thank you for, the engagement, for the reconnection, for the opportunity to be part of, this, really, sophisticated and, well-versed podcast series in cell and gene therapy, something that I'm really passionate about, and I know that you are as well.
I thank you. I certainly am. And I share your sentiment. And I'm happy we're able to you're a pivotal voice in cell and gene therapy. So I'm happy you're with us. And we're going to talk all about the impact of the patient journey as it applies to the cell and gene therapy clinical trial, which is an extremely important topic and one that we don't necessarily cover all that much, if at all, on Cell and Gene, the podcast. So what I want to start off by talking about is when you yourself step back and look at the full cell therapy trial journey, where does the patient experience today, experience the greatest friction? And what stages would you say are most ripe for meaningful simplification for the benefit of the patient?
Yeah, it's a great question, and I think it's absolutely pivotal, Erin. We always have to remember that the patient has to be at the epicenter of everything we do and everything that we say. And that's certainly been a very clear message, from me as I sort of think about my personal experiences in cell and gene therapy, starting my career in bone marrow and stem cell transplantation, taking that experience and then moving into biotech and biopharma and really developing these highly complex therapeutics. I think when we zoom out and we sort of just almost kind of examine the entire cell therapy pathway from patient diagnosis right through to long-term follow-up, I think to answer your question, the areas of greatest friction I think are rarely confined to a single step. I think that they occur in transitions. As I think about the patient journey, the referral stage, the manufacturing interval, when we think about autologous CAR-T, for example, the post-infusion monitoring phase. So I think if we kind of segment it, the first friction point is, in my mind, access and referrals. So many patients are eligible for advanced therapies, but these patients may not be treated at major academic centers. They may be managed in community settings where awareness of ongoing trials may be limited, for example. So I think we need to kind of focus on awareness and education. By the time a referral is made, I think when we think about some of these disease entities, the disease may have progressed. Eligibility criteria may have narrowed, or even the patient's clinical status may have changed. So I think, the delay is not just an operational one, it's quite emotional for the patient. And patients and families often describe this early part of this referral process as being very opaque and slow, particularly when, for some patients, time is critical, such as in the relapsed hematological malignancy setting. So I think that's one thought. I think the second, and perhaps maybe most distinctive friction point in cell therapy, is the vein to vein interval, particularly, I think, again, in the more complex scenarios such as autologous treatments, from apheresis to manufacturing to release testing. I think this period can feel like a prolonged holding period for patients. Again, there's a lot of anxiety. The patient is in this holding pattern almost. So I think whilst these processes are scientifically and regulatorily essential, for patients, they represent a degree of uncertainty. They're waiting while the disease biology continues to evolve, because cancer, for example, is very complex, and can be very transformative in terms of that evolution of the disease. So I think reducing that vein to vein time, improving predictability of things like manufacturing slots, and providing really transparent communication checkpoints during production, for example, could dramatically improve patient confidence and reduce anxiety. Again, really thinking about how do we put ourselves as drug developers in the shoes of the patient, and really kind of take that journey. And then the third friction point in my mind is long-term follow-up. So we know that gene-modified therapies require an extended monitoring, often spanning many years. Again, for patients who may feel clinically well after an infusion, repeated visits to specialized centers can feel burdensome. If we think about travel and time off work, caregiver logistics, depending on the scenario, cumulative fatigue is often underappreciated. So again, this is where we need to kind of think very creatively in terms of things like hybrid monitoring models. If a patient's been in a clinical trial, for example, or maybe integrating local laboratories, there's a focus on being much more innovative in terms of digital solutions, so using telemedicine and remote data capture. I think all of these offer a very meaningful opportunity to really simplify the patient journey somewhat without compromising regulatory rigor, because this is a highly regulated environment. So ultimately, I think simplification is most needed at these transition points for the patient, particularly where operational complexity directly, I would say, intersects with that patient vulnerability. And that's, I think, what we need to really pay attention to.
Agreed. All 3 points, spot on, extremely insightful. And what we hear certainly on Cell and Gene, what you're saying corroborates what a lot of our subject matter experts talk about there too. And I think I want to dig a little bit deeper into what that simplification across the trial design and logistics looks like. And so we know that early decisions around trial design, logistics, clinical supply strategy, they happen before the patient is enrolled. And so how do those upstream choices ultimately shape the patient's experience once they do enter the study, wherever they may be geographically?
Yeah, again, I think it's a really great question. And again, very pertinent. I think these upstream decisions in cell therapy development are, in effect, somewhat architectural. This is the framework, the practical framework, or the infrastructure that we build around the patient or around the patient journey. So once the patient is enrolled it's often too late to correct structural burdens embedded in the protocol, or again around that framework that we're building around the patient and the patient journey. I think that eligibility criteria, and that's a prime example. So whilst we can be very scientifically justified, can be overly restrictive, and the criteria potentially can exclude patients who are clinically stable enough to benefit. So moreover, when we kind of think about, again, that, that sort of overarching criteria, complex screening algorithms requiring multiple imaging studies or repeated biopsies or extensive lab panels. Again, let's think about putting ourselves in the shoes of the patient. This adds procedural burden. And this is before a patient even begins therapy. So I think each additional requirement increases our risk of attrition. But for the patient, this is also a psychological fatigue. When we talk about manufacturing, and manufacturing strategy also profoundly shapes the patient experience. So as we'd mentioned earlier, in the autologous model, centralized manufacturing may provide quality consistently, but it can limit this geographic flexibility. So if we kind of bring that back to kind of the site and the patient experience, you think about slot allocation systems that are not synchronized with disease progression or the dynamics. And this can create, I would say, both ethical and logistical dilemmas. So again, these are intricacies that need to be highly conjugated to that patient journey. But conversely, I would say that a decentralized or a regional manufacturing hub may improve responsiveness, but maybe introduce additional coordination challenges. So these are strategic decisions. They're not merely technical. And I think they determine how long a patient waits, how far they travel, and how predictable their treatment timeline actually feels. And we mentioned earlier about the anxiety that the patient feels during these windows of time. I think the other thing I would mention is, and very similarly, cold chain design and supply chain integration, again, influence reliability. For example, a cancelled infusion due to a courier delay or documentation discrepancy, again, it's not just simply an operational event. It's a deeply destabilizing moment for a patient who is prepared emotionally and physically for the treatment. So in many respects, operational design is patient-centered care, but with some degree of nuance. And when we thoughtfully align upstream decisions, they can create a seamless journey. But when it's misaligned, they amplify friction at precisely the moments when patients are most vulnerable. And I think that's why coming together as key stakeholders within the ecosystem to really ensure that we are focusing on the patient journey and influencing it as positively as possible, that I think is what's going to help us really scale and bring these transformational therapies to patients with real benefit.
Would you say from patient qualification through onboarding and consent, what are the changes that would most improve the clarity, the speed, the trust for patients navigating, as you've already outlined, a very already complex clinical trial process?
Yeah, it kind of, reminds me of my personal experiences when it comes to designing trials, designing, that framework, as I'd mentioned, the infrastructure that we need to build around the patient. I mean, cell and gene therapies are among the most scientifically complex interventions in modern medicine. I think the treatment effects are irrefutable, yet patients must understand and consent to these therapies. And that usually occurs within a relatively short window for some of the reasons that I previously shared. Again, I cast my mind back to my time in bone marrow and stem cell transplantation, the very early days of immunotherapy as we know it today. So improving clarity and trust begins with rethinking in some ways how information is delivered. I think certainly in my experience, consent documents are often very dense, they're very technical, they have a regulatory tone. And again, whilst completeness is essential, comprehension is equally critical. So it's finding that balance. I think a layered consent approach combining maybe written documentation with visual summaries, for example, or structured discussion sessions and opportunities for repeated questioning. I think that can significantly enhance understanding, and patients should not feel rushed into consent. Rather, they should feel progressively informed. I think being a chairman of a patient advocacy group in the rare disease space, for me personally, it's about how do we develop those tools and those opportunities to engage and to help amplify the patient voice. And I think even having dedicated patient navigators that could be another really transformative intervention. A single point of contact who understands the protocol, who coordinates appointments and facilitates travel support, answers logistical questions, and reduces fragmentation in that part of the process, which is really critical for the patient. Mm-hmm. And in complex therapies that involve apheresis or conditioning regimens or inpatient monitoring, long-term follow-up, this continuity of communication is foundational to building the trust of patients. So in some ways, speed is also crucial, as we'd mentioned before. Delays in screening approval or even things that we take for granted, insurance verification or scheduling, that creates distress and anxiety. So having, I think, clear milestone-based timelines, even if they include some level of contingency, because unfortunately things do go wrong, that helps to manage patients' planning and their expectations. So transparency about what is known, what remains uncertain, and what may change builds credibility with the patient. And ultimately, I think that, and we focus on this because it's so imperative, but ultimately trust is established not solely through this scientific excellence, but through consistency, clarity, and responsiveness. Certainly my personal experiences.
Yes, I think that's incredibly insightful and important, and especially to the patient and the patient's caregivers, where, for most patients, I think the science is, and the complexity of their condition is, for most of lay people, it is unheard of. And so the communication and the thoughtful, consistent communication, kind of by, like you're suggesting, one point person, is incredibly thoughtful. It's incredibly meaningful. And if you can take out the uncertainty and manage expectations, it not only prepares the patient and the families for what's coming, but also just eases a ton of stress, which you explained thoughtfully in your, just now. So that might be, in addition to curing the disease, helping to, cure the disease itself, hopefully, just managing expectations and putting the humanity into this is just tremendously important. And I think everything you outlined is incredibly important and hopefully certainly doable for any of the companies doing this. I want to talk a little bit about sample and data collection that involve multiple handoffs across sites and labs and logistic partners. So how does standardization and integrated transport actually help reduce delays and create a more seamless experience for the patient?
Yeah, I think, Erin, to your question, this is really foundational, isn't it? This is really the, the sort of the foundation of cell-based approaches, for example. I mean, cell therapy trials are uniquely dependent on this ability to kind of think about precision logistics. Biological materials may travel from a clinical site to a central lab to a manufacturing facility and then back again. So each transition carries potential, unfortunately, for delay or deviation or even error. I think as an ecosystem, we've certainly made some really significant strides in how we think about standardization through harmonized SOPs or validated packaging kits. So things like real-time temperature tracking and a unified digital platform. These steps that are implemented all reduce variability. And I think that even on the practical side, again, certainly in my experience, when we think about integrated transport partners who operate under the shared visibility systems, which allow sites to track shipments and anticipate arrival times more accurately. I mean, this predictability prevents things like rescheduling and reduces the likelihood of repeat procedures, as we'd mentioned before. So again, we're tying the practicalities and the nuances and this attention to detail from a system perspective right back to the fundamental, which is the patient journey. So from a patient's perspective, these efficiencies translate into fewer needle sticks, shorter visit durations, and lower risk of last-minute cancellations. As I'd mentioned before, a failed sample shipment may require a repeat apheresis or additional blood draws, so procedures that are physically taxing and emotionally discouraging for some patients as we think about that journey. So moreover, standardized data capture ensures that even adverse events and laboratory results are communicated consistently and promptly. And I think when these systems are integrated, clinicians can respond more rapidly, for example, into emerging safety signals. And I think, again, that reinforces patient confidence because we're being much more upfront with patients. We're keeping them informed. We're focusing on that level of awareness and sharing. So I think operational integration is therefore not simply about compliance. We talk about quality and compliance, and that's absolutely key to success. But it is about preserving patient dignity by minimizing avoidable disruption in that patient journey, I would say.
Excellent. And now I want to talk a little bit about long-term follow-up, which we know is essential in cell and gene therapy. So how should sponsors and sites rethink the monitoring and the communication that goes on post-infusion to support patients while they're still meeting all of these rigorous clinical and regulatory requirements?
Yeah, I mean, long-term follow-up is both scientifically a necessity, and I think to a certain extent, it's a moral responsibility. Again, I think about my experiences in the bone marrow and stem cell transplant setting where we were building long-term survivorship programs. This is a very, very similar framework, because we know that gene-modified therapies may carry theoretical risks of delayed adverse events or more sophisticated components such as insertional mutagenesis or even some unfortunate loss of efficacy over time. However, I think that, monitoring frameworks must evolve to reflect the realities of the patient life. And I think, as you'd mentioned in your question, hybrid models are increasingly very viable. And mentioned earlier about, telemedicine consultations or remote electronic patient-reported outcomes, wearable biometrics, and even partnerships with local laboratories can I think significantly reduce the travel burden for patients. But at the same time, regulatory requirements must be and can be satisfied whilst allowing patients to resume, I think, normal routines. And I'd mentioned kind of the post-infusion part of the patient journey and what that means in terms of trying to minimize the burden on the patient. So I think communication, even during long-term follow-up, should also shift from episodic to continuous. We talked a little bit about that engagement with the patient and what it means to continuously share information. So I think patients benefit from these regular check-ins, educational updates and clarity regarding how their data, for example, contributes to our broader scientific understanding. Because I think when patients understand the purpose of the overarching kind of monitoring algorithm, it's not merely the obligation. I think they're more likely to remain engaged because there's a reason to believe that there is a purpose. So I think psychosocial support, of course, should not be overlooked, because after receiving a potentially transformative therapy, patients still live with a degree of uncertainty regarding things like durability. And having, I think, spoken to a number of patients who are post-CAR T, for example, it's certainly clear that that structured follow-up that includes mental and health and well-being resources, survivorship counseling, and that peer support can also mitigate this anxiety. So I think the future of long-term monitoring should be built around partnership, where patients feel not only observed, but supported for all the reasons that I've just shared.
Absolutely. Couldn't possibly agree more. And I think in that same vein of how helping patients to feel not observed, but supported, there is risk involved in everything that we're talking about. And so how are knowing that patients and families are increasingly aware of high profile safety stories in gene therapy, how should sponsors and investigators discuss risk in a way that's honest and open and thoughtful, but not paralyzing and, just completely overwhelming the patient?
Yeah, no, it's a great question, Erin, and I think it's one that requires us to be very methodical, very thoughtful, to take a very balanced approach. I mean, we should assume that patients have seen headlines, even if they don't raise them explicitly during a conversation. And as you'd mentioned, there certainly has been reports in the public domain, but we need to kind of keep it in context. So I think in my mind, the right approach, as I'd mentioned, is to contextualize the risk without minimizing it, but explaining what is known, maybe what's uncertain, but also to the previous question, what monitoring is designed to catch early, what symptoms require immediate attention. And I think our ability to be much more predictive around the tools that we're developing, the application of scientific rigor, even AI and machine learning, I think is allowing us to really be much more critical. And I think that's what's really key. I mean, critically, risk communication should include the practical lived experience, how monitoring will affect daily life. For example, the frequent labs, early post-infusion, what proximity to care might be recommended and how caregivers can support. So I think, again, it goes back to the fundamental. When patients feel informed and supported, rather than just being sold to, that increases trust, even in this face of uncertainty, as we continue to garner more experience or more real-world experience with cell and gene therapies. But as I'd mentioned, the key here is to ensure that we're being very thoughtful, very methodical, and we're contextualizing the risk.
Absolutely. Absolutely. And before I let you go, I have one final question for you. So we know there's a growing emphasis on real world evidence after approval, especially for long-term outcomes in gene therapy. So how should patients be or can be engaged in that shift?
Yeah, no, I again, another great question, Erin. I think that, as I just kind of reflect on, the conversation and the great questions that you posed, again, I think there's a common theme here. I think that patients shouldn't feel like a data source. I think they should feel like partners in building the field's knowledge base. And I think, the direction of travel is very clear. Patients are our North Star. And I think long-term follow-up increasingly intersects with real-world data systems, registries, for example, to understand durability and late safety signals and outcomes across broader populations. I think that there are organizations out there that are collating this real-world data, looking at trends and patterns and signals. And I think that only works if participation is practical, it's respectful. So again, it goes back to the first question that you posed: minimal friction reporting tools, very clear consent about how data is being used, feedback loops where patients learn what the community is discovering, and support for the burdens that come with prolonged monitoring. And I feel that, certainly in my experience, if this is done properly, this creates a virtuous cycle, which leads to better evidence, informing better guidelines. And again, I think that has a knock-on effect, more confidence from clinicians, from regulators, and from payers. And this is all going on within this very complex ecosystem with many key stakeholders, but this all preserves patient dignity and autonomy, in my personal and humble opinion.
Well, thank you. And listeners, at the top of our discussion, I explained that Panteli is a pivotal voice in the cell and gene therapy sector, and evidenced by our conversation today, you can absolutely see why. And he lives literally at where complex lab science and real world diagnostics and deployable cell and gene therapies converge, and we're turning experimental concepts into safe, testable, scalable treatments for patients in a thoughtful, human way that puts the patient first. And so, Panteli, I can't thank you enough for your time. This has been incredibly insightful, thoughtful, and you're welcome back to talk to our audience at any time, especially as it applies to putting the patient journey first.
Erin, again, thank you for the opportunity. Thank you for your time as well, for your thoughtful questions. And it's really been, a pleasure and an honor to be part of the podcast today under Cell and Gene. So again, thank you very, very much indeed.
Absolutely. Thank you. And I hope we get to see each other at a conference sometime soon, whether it be in the UK or here in the United States. So that's something to look forward to too. But listeners, thank you as well for your time. Please visit Cell and Gene at cellandgene.com and subscribe to Cell and Gene, the podcast, on any major podcast platform or wherever you listen in. And we will talk to you soon.